Alright, in this video we're going to talk about the cell cycle. So this is kind of like the life cycle for a cell. So we're going to start off, here's our cell. That's our nucleus, and it's going to start off with chromatin, okay? Our DNA is all spread out and squiggly in the nucleus. So when it looks like that, this cell is going to be an interphase. Okay, it's going to be an interphase. And there are three parts to interphase. So we can actually break this down. This is going to be G1, S, and G2 phases. Alright, so in the G1 phase, the cell is going to grow. We're going to remember G stands for growth. We're also going to make more organelles. So we're going to grow and make more organelles. Now remember, the whole point of interphase is to get ready for that cell to divide. So throughout the cell cycle, what's going to happen? It's going to live most of its time in interphase. Alright, then this one cell is going to start to pinch in, and it's going to become two different cells. So one cell is just going to pinch in and become two cells. So we need to make more organelles so that each cell has enough mitochondria or enough ribosomes in each cell so that cell can function. All right, our S phase of interphase is going to copy the DNA. Okay, it's going to copy the DNA. So once again, same thing, right? We have one cell. It's going to split into two cells. If we only have one copy of the DNA, that means this one's going to get it and this cell will die. Okay, so that's going to, S phase, we copied the DNA. All right, in the G2 phase, we're going to grow some more. We can remember this by G standing for grow. And it's also going to double check and make sure that we copied that DNA right. So double check that DNA. Okay, we got to double check that DNA. All right, and that makes up interphase. So like we said before, most of the time a cell is going to spend its time in interphase. All right, the next stage is called prophase. And there are three things that happen in prophase. So the first thing that's going to happen is the nuclear membrane. So the area around the nucleus is going to start to dissolve. Okay, so we're going to draw it dotted because it's starting to dissolve. We're also going to see centrioles appear. So centrioles, we're just going to draw them as little t's. But those, if you think back to our organelles unit, they help with cell division. So now we're talking about cell division. Okay, and then the last thing, we are going to have some chromosomes. I'm going to do, we'll do 3 chromosomes. I'm doing different colors. Okay. So let's think about, just ignore that mark. Okay. Let's think about what just happened. So the nuclear membrane dissolved. Nuclear membrane dissolves. Centrioles appear. And our chromosomes condense. So it was that chromatin, but that chromatin is going to condense. It's going to coil up and tighten into chromosomes. So we'll just say chromosomes condense. All right, those are the longest two ones. The rest of these go by pretty fast. All right, so after prophase, we have a couple other things. So you may have heard of PMAT. So this is where the PMAT is going to come in. So PMAT, PMAT. And these PMAT steps are the steps of mitosis. So this whole thing right here, this is all part of mitosis. And so this is when the cell is actually reproducing. This happens very, very fast, even though there's a lot of steps. Most of the time, like we said, a cell is going to spend its time in interphase. All right, with mitosis, we just remember PMAT, so we did prophase. The next thing that's going to happen is our chromosomes are going to line up in the middle. So 1. Okay, and then don't forget, we also have the centrioles on the side. Okay, so this is metaphase. And for metaphase, the chromosomes are in the middle. It's easy to remember because metaphase, middle. The next phase is anaphase. So in anaphase, our chromosomes are going to be pulled apart. So those sister chromatids that were together are now going to be separated. They're going to be ripped apart. So we still have our centrioles on the side. This time, we're drawing my spindle fibers. Those sister chromatids got pulled apart. So, sister chromatids pulled apart. Good, and then the last one is telophase. We're going to remember this one because it kind of looks like an old school telephone with the circle part and then the bar that you just like hold on to and then the other circle part. I know you're going to have to use your imagination, but it's going to pinch in right here. This is called the cleavage furrow. So you can tell between here and here, it's starting to pinch in. And then also for anaphase, make sure you know apart. Anaphase apart. All right, going back to telophase. So we're going to have 2 nuclei up here. So then the nucleus is going to start to be formed back. And then the 3 chromosomes are going to be in each one. Okay. So the 2 things that happened here was cleavage furrow appeared. And the 2 nuclei formed. This is kind of interesting because technically there's 2 nuclei in the same cell. All right, and then the last stage is when our cytoplasm separates completely. This is called cytokinesis. And that's just cytoplasm separates. So we still have the same 2 nuclei and each color chromosome in each one. And so now we have two cells. So remember, we started with interphase. Most of the time cells spend their time in here. Then mitosis happens. So we just remember mitosis by PMAT. And then last we have cytokinesis, which is when the cytoplasm separates completely.